Hello, Mr. Allman here, and this is your Allmancast on the regulation of gene expression. Genes are short sequences of DNA, which, when expressed, are transcribed and translated into a polypeptide sequence. Now, we have tens of thousands of genes and nearly 100,000 different proteins. It would be wasteful to expect that every cell is going to produce every one of those proteins at all times. It is for that reason that regulation must occur. Regulation are going to be the processes which are going to control which genes produce which proteins at which time in which cell. All of these things are going to allow for a cell to be more efficient, and it's going to allow for different types of cells to be able to accomplish their different functions, have their different structures and shapes, in order for them to be the cell type that they were meant to be. Now, eukaryotic cells, because they undergo transcription and translation in different parts, the uncoupling of that allows for there to be more steps, more pauses during which regulation can occur than in comparison to a bacteria cell, which is accomplishing transcription and translation simultaneously. This all starts with DNA, particularly access to DNA. In order to ensure that the correct proteins are made at the correct time, DNA that should not be transcribed can be made inaccessible. There's a couple of ways in which this can be accomplished. DNA itself can be modified with the addition of something called a methyl group, which is simply one carbon and three hydrogens added onto the surface of the sugar phosphate backbone. When a large number of those methyl groups are added in a particular spot on the DNA, that region of DNA is now going to be inaccessible to RNA polymerase, and transcription is effectively blocked. Those methyl groups can be present at birth, or they can be added or removed due to environmental factors over the course of one's lifetime. Additionally, if a sequence of DNA is wound around histone proteins, then that DNA, that region of DNA, cannot be transcribed. So DNA that is within the histone is inaccessible, while sequences of DNA in between histones remains accessible and can be transcribed. The patterns by which different regions of DNA are methylated and wound or unwound is actually heritable, and so changes that occur to one's DNA during one's lifetime can thus be passed on to future generations, meaning that the beneficial or harmful effects of lifestyle choices can then get passed on not only to the offspring, but to many future generations, because those changes can be encoded in the DNA itself. An interesting application of this DNA access is seen in these transcription factories, because chromatin is clusters of DNA clusters of nucleosomes of DNA. Transcription factories are places where loops of those chromatin can come together in areas where transcription increases. And so this allows for very common genes, proteins that are needed in large quantities, can then more efficiently be produced in large quantities in a short period of time. Before transcription occurs, regions of DNA above stream are going to control for whether or not a particular gene should get expressed. There are many sequences of DNA. Enhancers and promoters are two words that you will hear often that describe sequences of DNA to which particular proteins need to bind in order for RNA polymerase to actually begin transcription. Collectively, transcription factors are the proteins which will bind to those different regions of DNA. Now, sometimes they can actually appear a very large distance ahead of where the gene occurs. In this case, we have enhancer regions. Those enhancer regions require the binding of very particular proteins these proteins will attach, lead to a bend in the DNA, recruit a group of general transcription factors, which will in turn recruit an RNA polymerase to be able to bind to the promoter region, which will then allow for RNA synthesis to begin. Even if the transcription factors and the RNA polymerase are around, without those mediator proteins attaching to those enhancers, the gene will never be transcribed, and therefore that protein will not be produced. Therefore, the presence of these mediator proteins that are specific to those enhancers is then important to increase the rate and the likelihood of transcription actually beginning. This is going to be important in allowing for differential gene expression, because depending upon which cluster of available activators are there, that will determine which proteins will be made. We can see here on the left we have a liver cell which has a set of activators available within the cell itself that lead to the expression, for example, of albumin, but the correct set of enhancers, activators, is not there to trigger the production of crystalline. Albumin is a gene which will be important in the liver. Crystalline is not something that is necessary in a liver cell. On the other hand, we have a lens cell in the eye. A lens cell does need crystalline. It needs this particular protein, and it will have present in the nucleus the activators necessary to bind to those enhancer regions of the DNA, and trigger transcription of that correct gene. So the differentiation of different types of cells will be influenced depending upon which of these factors are present in the nucleus of those cells, and therefore lead to the expression only of proteins that are necessary for that cell's function. Moving ahead, after transcription, when you have more options in terms of different combinations of introns and exons, then that's going to allow for different outcomes in terms of results. We have the 5' GTP cap and the 3' poly A tail added onto either end. And as that's happening, we have introns and exons which can be spliced in order to produce different combinations of proteins as a result. The vast majority of human genes with exons and introns that we are aware of will undergo this process. You can see this gene given as an example, which has 5 different exons in it, could be spliced in one of those 2 different ways. We could put together exon 1, 2, 3, and 5 to make mRNA strand number 1, or we could put together 1, 2, 4, and 5 to make mRNA strand number 2. If we look, the sequences present in here is going to be different in those RNA bases. Therefore, we're going to get a different amino acid sequence. Different amino acid sequence leads to a different shape, and therefore a slightly different or modified protein, which will then have a different function. This is what allows for there to be multiple different proteins produced from the same set of DNA. During translation, in order to, instead of prevent or stop the process, there is a way in order to increase the rate at which translation and thus protein synthesis is going to occur. And this is a structure called a polysome. A polysome is a cluster of ribosomes that are all bound to the same mRNA strand. And this is a process that is going to allow for an increase in the efficiency of a particular type of protein. In the electron micrograph, you can recognize polysomes when you have a cluster of little circles or spheres all bound to a thin thread. That thin thread is the strand of mRNA, and each dot on there represents a ribosome. The ribosomes that are closer to the 3' end of the mRNA those are the ones that have had a longer period of time to produce the protein, and those closer to the 5' end, where the reading of mRNA begins, are going to have a shorter strand of that growing polypeptide. As that mRNA passes through, beginning with the 5' end entering in this particular diagram from the right going towards the left, the more ribosomes it passes through, the more copies of that particular protein are produced simultaneously from one piece of mRNA. That allows for a reduction in the number of transcripts that need to be produced by the nucleus for that gene, because each strand of mRNA can be used multiple times. Finally, after translation has occurred, proteins that are not needed need to be broken down, and so regulation can occur by the quick breakdown of proteins that are no longer necessary as a way to then be able to recycle those components as individual amino acids to be used to be able to make new proteins in the future. And that is a brief outline of regulation of gene expression in eukaryotic cells. Thank you for watching.